This is our first lecture in Bio 100. We're going to be talking about biology as a science and therefore what is science and how is science conducted? What is the scientific method? Science is essentially our attempt to understand the natural world. And we try to do that with information that can be observed and can be observed by more than one person. So with information that, in a sense, others could verify. Science is pretty important in our economy. The technology that you're using every day would not be here without a lot of great leaps in scientific knowledge, chemistry, physics, lots of things. And of course, all that you are, you're a living organism, so biology is going to be very important to you. The U.S. is not the best in its performance at teaching its citizen students how to know the ins and outs of the scientific world, but we are getting better. We do have a good, strong economy, and there are many jobs that will require people that fill them to have some knowledge of science and sometimes very high levels of proficiency in some sciences. So that's why we want everybody to have some science when they're going through getting a degree at MCC. Biology is the science we will be studying. It is the science of life. That encompasses anything that happens within what we call the biosphere, that very thin film of life that covers the planet. And in a sense, we will be looking at a very small portion of all the things you could study in biology, but we'll try to be looking at a broad spectrum, and hopefully you can find some interest in many of those places. The properties of life, if we're going to study life, are that it's order. It has order versus the more chaotic environment in which you find it. It grows and develops and regulates its environment, its internal environment. So a cell has to regulate its chemical properties. It has to regulate energy coming in, waste going out, etc. And then life grows and develops, changes, and reproduces. It uses energy to do this. Energy is required to maintain order against the chaos in which it finds itself. And we find that life changes over time. As it reproduces itself, there are occasionally changes that occur in each form of life. And over time, that has generated what we call evolution and a great biodiversity on the planet that we can study. So when you look at life, there are different levels at which you can study it. You can literally study just the chemicals that go into building life. But the fundamental unit of life, we'll see, is the cell. All life exists as one or more cells. Cells can become organized into tissues within an organism, and those tissues can become specialized into organs. So your muscle tissue is different from your liver tissue, etc. The organs together are components of an organism, a unit of reproduction, as it were. And then organisms are occasionally organized in populations, depending on how we define a population. Populations that are found together in the same area, the same kind of habitat frequently, are referred to as a community. And you can study specific communities like the desert community, the Sonoran Desert community that we have here. Multiple communities, if you look at a large enough sector sort of the planet, you're getting into an ecosystem. A community tends to be all of the organisms, all the different types of living things and how they interact in an area or in a habitat. Whereas when you get to an ecosystem, then you're not only talking about all of the organisms, all of the different species in an area, but all of the non-living things, what we call the abiotic components of that environment, the rock, the soil, how much water comes in the rain or how much flows from a stream, sunlight, anything that is non-living that is in that system is abiotic, and it's extremely important to the organisms within the community. So if you're looking at the organisms and those abiotic factors, you're looking at an ecosystem. And then finally, if you slap all the ecosystems together and you look at them, that gives you a view of the biosphere, which is that thin layer of life, that very thin film of living stuff that exists on the planet. When we conduct science, we try to do it in a way that protects us from a couple of things. One, we want to be able to believe that our understanding, the understanding we're getting by conducting science, is a good understanding, that it's accurate. So we want to have accurate data, accurate information. On the other hand, we also know that we are biased as human beings. We are not perfect, and we will tend to let our biases affect how we perceive information, how we collect data even. And so the scientific method is a method to sort of keep us as honest as we can be about conducting science. Scientific method is hypothesis-driven science. A hypothesis is simply an explanation, a proposed explanation of something that you've observed, some phenomenon. So the components of the scientific method, and we'll have a few examples of this, are as follows. You have an observation. Okay, we're studying the natural world, so you've observed something in the natural world, like the fact that a number of butterflies out there are black and orange in color. And orange is a common butterfly color amongst certain groups of butterflies. All right, so you've got a causal question then that comes from that. Well, why are these butterflies orange and black? Is there some reason why they're orange and black? Not sure. But then you generate alternative hypotheses. Basically, these are potential answers to that causal question. And I won't go into the black and orange butterflies any further right now. And we'll just keep going with this process, and then we'll use some examples in just a moment. You want to have multiple working hypotheses for whatever observation you're trying to explain, because you don't want to get stuck on just one possible explanation. In a sense, that is letting your bias control too much of how you're conducting the science. Once you've got your hypotheses, then you've got to try to figure out how you would distinguish between hypotheses that seem to explain the phenomenon versus hypotheses that don't. And so you need an experiment to ferret that out. We call that an experiment or a test. So you're testing these alternative hypotheses. You design the experiment, and from the experiment you generate predictions based upon the assumption that each of your alternative hypotheses is true in turn. So if hypothesis 1 is correct and I do this experiment, X should happen. But if hypothesis 2 is correct and I do the same experiment, then Y should happen. You generate these predictions before you really do the experiment, because if the predictions all end up being the same, they all predict X, then your experiment stinks and you need to redesign that before you go do it. Once you've got a good experiment and you've got your predictions in place, now you know you shouldn't be biased about collecting the data. You've got a clear way to collect the data, and you've got something clearly to compare the data you collect to, your predictions. So you go collect the data, you go do the test. Once you've got that information, those results, then you can conclude. You can see whether any of your hypotheses are supported. So you compare your results to your predictions and see if the data supports or matches a prediction and therefore supports one of the alternative hypotheses. This is not rocket science. Science is not just for smart people in lab coats and chemical laboratories or something. This is something that we do. So I'm just going to put sort of a, hopefully not everyday event into this context of the scientific method, but a common occurrence into it, something you may have run into in your life. I go outside, I go to get in my car and go to work, and I get in the car, I try to start the car, and the car won't start. So that's my observation. The car won't start. Well, with a few choice words, probably, I will eventually ask the question, why won't my car start? Maybe not out loud, but I will ask it. And there's many reasons why your car might not start. You can go on YouTube and look up all the possible hypotheses for why your car won't start. We'll just use a couple of them here. I've got hypothesis 1 as being that I'm out of gas, and then hypothesis 2 that my battery's dead. So if those are the two hypotheses I've got, and I need an experiment to ferret out whether one or the other is supported, I need an experiment, a test that will be based upon at least one of these specifically. So my test is going to be that I hook up jumper cables to my car and the car battery as you're supposed to, and then to someone else's functioning car battery. And then you go try to start the car. So you're basically bypassing your battery and using the other battery to turn your engine over. So you're bypassing your battery that may be dead with a functioning battery from a car that's generally running, so you know that battery's good. All right, so my predictions, assuming each hypothesis is true in turn. If my car won't start because I'm out of gas and I do the jumper cable test, then when I try to start my car, it still will not start. Bypassing my battery isn't going to do anything to put gas in my car, so that shouldn't solve the problem of the car starting if the reason why the car won't start is because I'm actually out of gas. Prediction 2 is based upon hypothesis 2. So if my battery is dead or discharged and I do the jumper cable experiment bypassing the dead battery with a good battery, then the car should start. So this is two different predictions, two different results. Hypothesis 1, the prediction is if I do this test, the car won't start. Hypothesis 2, the prediction is if I do this test, the car will start. Okay. So then I go do the test. I wake up my roommate or my wife or whomever and hook up the cars. I could probably do that on my own without bothering my wife. But in any event, if I do it and the car starts, then in the scientific way, you'd say, all right, well then hypothesis 2 is supported. My battery seems to have been discharged. And so then in the real world, you say, might be something wrong with my battery. I better go get it checked. And if you live here in the desert, you know that they never live their full warranty. They only live about 2 to 3 years. So that is the process of science. We do this as problem solving all the time. As science continues, a lot of the time we make our observations, we get our questions, we formulate hypotheses, we test the hypotheses. That may allow us to reformulate our experiments, refine them, because some of our experiments may not be good. And then eventually when we get a good experiment and we get some data, then we'll get some information, and eventually you're going to be getting more observations, in the sense that when you answer one question in science, very frequently that leads to other questions. And so you're continually building upon your knowledge and understanding of the natural world. All right, let's look at a biological example of a scientific method. Here are two snakes with banding patterns that involve black, red, and sort of a cream yellow or yellow. And one of them is a harmless king snake, and the other is a coral snake. So there's one that's non-venomous and one that's venomous. And it's important to know the difference. If you don't know the difference, don't pick up any snakes that look like this. But imagine you were a predator that eats snakes, like there are certain birds of prey that will eat snakes. You might want to know when you would be able to eat a snake and when you wouldn't. One of the questions, though, would be, if one of these snakes is venomous and one is non-venomous, why do they both have such similar patterns? And a hypothesis to explain that is that the venomous organism develops brightly colored pattern, and that's a warning to predators that it's poisonous, it's dangerous. It advertises that you should not try to eat me or kill me because that's bad. If something can learn that or understand that it's bad to attack you before they attack, you both win. And of course, as an organism that might get attacked, you might really only care that you win, but it's important that you do. Aposematic coloring basically is having coloration that warns predators that you are dangerous. And here we've got a couple of examples of that. We've got the monarch butterfly, which is black and orange. It eats milkweed. Milkweed has toxins in it. The larvae build the toxins up in their tissues, and then the adult, after the caterpillar turns into an adult, retains those toxins in its tissues. So if a bird eats a monarch, it will tend to get sick and vomit. It's a bad thing. So the monarch advertises that it's bad. And we see down here at the bottom 4 different bugs that are black and orange. That's a common combination to warn predators that you are potentially dangerous. Bees, if you see bees, black and yellow is also a common aposematic coloration. Rattlesnakes are black and yellow. So there are certain patterns and certain color combinations which have tended to develop and warn predators that the potential prey item is dangerous. We also see a poison dart frog here that exudes toxins in its excretions from its skin, and the bright colors on the poison dart frogs are an advertisement. They're aposematic coloration also. All right. So there's another way to avoid being eaten, and that is to be cryptic, to have your coloration allow you to blend into your environment and your background. And we've got a couple examples of that. We've got a parrot that actually looks a lot like an owl, but it's flightless, and it blends into the green background of the leaves in the forest where it exists. We've got a lizard blending into the rocks where it is, and a butterfly whose wings look like the dead leaves on which it is perched. So that's another way to prevent yourself from being eaten, is to prevent yourself from being noticed at all. And that is to be cryptic in your coloration. So an experimenter wanted to test the aposematic coloration hypothesis with the coral snake, and went down into Costa Rica and used coral snake patterns and typical brown snake patterns to conduct a test of 3 different hypotheses. And the null hypothesis was that birds attack all snakes equally, doesn't matter what they look like. If they see a snake, they're going to attack it. So nothing that the snake can do, nothing that it looks like is going to make it more or less likely to be attacked by a bird. Hypothesis 1, H1 here, is that birds recognize the coloration of 3 colored snakes and they avoid them, the aposematic coloration hypothesis, so that if they see a tricolored snake, they're less likely to attack it than a brown snake. And the third hypothesis, but H2 here, is that birds don't notice tricolored snakes, i.e. they are camouflaged. The tricoloration camouflages them, makes them cryptic. So the experiment was essentially to put these snakes on a natural background, both the brown and the tricolored snakes, and then put the snakes also on just a plain white background, and record how often avian predators took interest in either type of snake. So, if our null hypothesis is that the rate of attack on these snakes will be the same for all snake colors, regardless of what surface they're on, then we would get data that looks something like this. The frequency of bird attacks would, 1, be equal between the tricolored snakes and the brown snakes, and 2 would be equal between how frequently they're attacked on a natural background versus a white background. The blending in hypothesis is not part of this hypothesis. Cryptic coloration would play no role. All right, under the aposematic coloration, our predictions would be that on the natural background, the birds are going to see a tricolored snake and not attack it very frequently because it's a warning color. Whereas if they see the brown colored snake, they'll attack it. And then if you put those same snakes on a white background, well, it doesn't change the fact that the tricolored snake is still tricolored, and that's aposematic coloration and a warning signal. And so therefore, they're still not going to attack the tricolored snakes on the white background, but they'll be happy to attack the brown snakes on the white background. Finally, if camouflage coloration, or the cryptic hypothesis, as it were the case, if that were true and we did this experiment, then cryptic coloration is supposed to make these tricolored snakes blend into the natural background. So if we put the snakes on the natural background, then the cryptic coloration of the tricolored snakes should lower their frequency of attack versus normal snakes. But then if we put them on a white background, since it's not a natural background, then suddenly the bird should be able to see them, and the attacks on the tricolored snakes versus the brown snakes should be about the same. So these are our 3 sets of predictions coming from the test we're going to do, or that was done, based upon these hypotheses. So each hypothesis generates a separate and easily recognizable different pattern of data, different predictions. So it's a good experiment. So what happened? Basically, no matter what the background was, the tricolored snakes experienced much lower frequencies of attack than the brown snakes, whether it was natural background or whether it was on a white background. So the null hypothesis that they should all be about equal, that's gone. The camouflage coloration hypothesis is gone because the rate of attack on the white background for the tricolored snakes, when they could obviously be seen, was still low. And so that leaves us with the aposematic coloration hypothesis being supported. So the results show that the coloration of coral snakes is probably due to the evolution of warning colors, aposematic coloration in this organism. All right, so this is simply how we conduct science. We conduct experiments that we design to be good experiments and generate different predictions based upon which hypotheses may or may not be, what we would call true. Technically in science, we never prove a hypothesis is true. We always hold the possibility out that we could be wrong about what we're thinking, that there might be some other explanation we haven't thought of to explain our observations, and that may be discovered and elucidated at some future date. However, it is difficult to talk about ideas and explanations and not get the idea that some of them are true, or some of them are certainly better than others. In any event, once multiple hypotheses or multiple tests, I should say, have supported a hypothesis, then you may raise the level of that hypothesis up to a theory and start talking about the theory of X rather than the X hypothesis. But again, like a hypothesis, you never prove the theory. You always keep it as a theory and essentially hold in reserve the possibility that you could be all wrong about this. Other sciences like physics and chemistry will tend to have laws because they are so predictable. There are certain patterns that matter takes in its interactions with other matter, etc., that you can, develop laws in chemistry and physics, say. And that's very difficult to do in biology. There is an exception, I can think of, which is Mendel's principles of heredity. Two of them were elevated to essentially Mendel's two laws, and those were segregation, the laws of segregation and independent assortment in cell division when cells are dividing to make what we call gametes, sperm and egg, pollen and ovules, reproductive cells. But otherwise, other than that, it's pretty unusual for a biological theory to be elevated to a law. All right, next we'll start talking about some of the unifying theories of biology. We will start with essentially Darwin's theory of evolution.